Have you ever found yourself thinking, I want a way to easily view a sequence position in my gene? For example, maybe you work with the human TP53 gene and you want to see coding position 586 in one of the transcripts. I'll show you two ways to do this in our Genome data viewer. The first uses the Exon Navigator widget, which sits directly above the graphical sequence viewer. I'm displaying chromosome 17, and I've already searched for the TP53 gene. As you can see, this gene has several transcripts. You can use the transcript menu within the Exon Navigator to select a particular transcript. I'm selecting NM000546.version6. Note that you'll only be able to choose transcripts from the latest NCBI gene annotation release. These circles in the Navigator represent each exon in the selected transcript. A mouse over on the exon circle provides information about the exon, including the exon number and the CDS range on the genomic record. The TP53 gene happens to be annotated on the minus strand of the genomic record, which is why the exon numbers decrease as you move from left to right. But you can always flip the strands, if you prefer, using the Tools menu. We're looking for coding position 586 in this transcript, so let's make a guess and start with exon 6. I'll click on that circle in the Exon Navigator, and it zooms directly to that complete exon. I'll hover my mouse in the purple transcript we are interested in, and a tooltip pops up, showing the exon number and positional information. I set my mouse over this G at mRNA position 737 and CDS position 595, so pretty close to 586. A quick tip. If you click on the transcript in the viewer, a ruler displays the mRNA coordinates, making it easier to mouse over the position you want. Here is the position we're looking for, CDS 586. A right-click with the mouse lets you set a marker at that position. I won't do that, but I will zoom in a bit to point out that you can easily see the amino acids in this region, and that a mouse over on the red protein object shows that our CDS position 586 is this arginine at protein position 196. Let's now look at the second way to find a position, which is to use the Tools Go To menu in the viewer. I'll open that menu and paste in an HGVS-like expression for our coding position in the transcript. I've pasted in the NCBI accession.version number, followed by a colon, then lowercase c, .586. And of course the bare genomic location would also work, 7674945. Finally, the last tip. You can download all of the exons for a given transcript from a link in the Download FASTA section of the tooltip for the transcript. To get the sequence for individual exons, use the Exon Navigator. Right-click on a circle, and you can copy-paste the sequence. We would love to have your feedback and questions, so please use the feedback link in the Help menu.